Right. Yes. Good morning to everybody. Today, yesterday, I discussed about the introduction about the photosynthesis. Now we will be discussing as the first one is the chloroplast, the pigments. Among the pigments, the most important one is the ultrastructure of chloroplast. Yes. See, this is the ultrastructure of chloroplast. The chloroplast you will be knowing. That is the most important character in the plant is presence of the chloroplast in the cytoplasm. So it is a cell organelle. It is a cell organelle. It is green in color because of the presence of the chlorophyll pigment. So it is a plastid. It is a plastid, but it is green in color because of the chlorophyll pigment. And especially in case of the chloroplast, the size of the chloroplast will varies from plant to plant and from time to time. Means as the plant becomes old, the cell becomes old, and the number of chloroplasts will be increased and the size of the chloroplast will be different. And moreover, generally, if you observe, it is ultrastructure means if you observe under the electron microscope, commonly because it is subcellular and also it is and it is very difficult to see with normal microscope because, and also here if you observe the chloroplast from the top view, it appears like a discoid structure, disc-like structure. If you observe from the side view, it appears like a oval-shaped structure, oval shape. And especially in the chloroplast, here it is about, the chloroplast will be about 4 to 10 millimicron in diameter. And also it will be 1 to 3 millimicron in the thickness, general. Here the chloroplast, the ultracellular, it is having two membranes. The outer membrane, from the side view, it is elliptical or oval shaped structure. The outer membrane, and the next one will be the inner membrane. In between the outer and the inner membrane, you will have intermembrane space, and then in the inner, inside the inner membrane, you will find a semi-fluid. We call them as, it is a fluid that is called as stroma or matrix. The stroma or matrix. So this stroma which will be covered, and then inside the stroma you will find the disc-like structures which are placed one above the other. They are called as granum. So these are called as grana. So they are called as grana. So these grana, they are placed one above the like the points. These point-like structures, they are called as thylakoids. These thylakoids, they are placed one above the other in a disc-like structure. So the entire individual, we call them as grana. The single, it is called granum. And these grana, they are interconnected with each other. And these grana will play a very important role during the photosynthesis. And also here, the chloroplast is having the starch grains in the stroma, starch granules, and also it is having the DNA and its own DNA, and also the RNA and the ribosomes. And in the stroma, there are soluble enzymes are present which are responsible for the dark reaction, and also along with them, there are some enzymes which are necessary for the synthesis of amino acids and also the fats. So these are present in the stroma. They are present in the stroma. And also these are matrix. And also these thylakoids, they are interconnected by a layer that we call them as stroma lamellae or stroma thylakoid lamellae or thylakoid lamellae or fret membrane. All are the same. These are the interconnecting membranes which connects each grana. And also here, the most important one is the DNA. It is just like it is circular in structure. The DNA will be circular like prokaryotes. About 1,35,000 base pairs will be there. In general. So it appears like it, and also it is having its own DNA and ribosomes. And these are the most important characters and of the chloroplast. And also here you will find that there will be three spaces we find this. The one space that is intermembrane space, The second space, that is called stroma space. The third space we call them as thallopoid space. So there will be totally 3 spaces will be there. So that is intermembrane space, then the stroma space where the stroma is present, and then between the thallopoid membrane. Thallopoids, they are disc-like structures, they are placed one more. So inside there will be the space will be there. That we'll discuss in later in the important functions of all these. And especially the chloroplast plays a very important role for the photosynthesis. And also here, the synthesis of the that is the one more important character here. These 2 membranes, they are having double lipid membranes. So these double lipid membranes, they are having lipid membranes. They are having some that is the transport proteins are present inside the membrane, and as compared to the outer and the inner membrane, outer membrane is more permeable than the inner membrane. Why? Because the outer membrane will have the more carrier proteins. That is the lipid membrane, the double lipid membrane, which is impermeable. And also there will be the carrier proteins will be there in the membrane along with the along the membrane. So these carrier proteins, they carry. So the outer membrane will be more permeable because the number of carrier proteins will be more as compared to the inner membrane. So this is about the structure of the chloroplast. And also here in the chloroplast you will find the 4 different pigments. That is the pigment. That is the one. The first one is the photosystem 1. The second one is photosystem 2. That I will explain you. The third one is that is the cytochrome complex. And the fourth one is cytochrome complex, and the fourth one is that is the ATP synthetase complex. So these are the 4 substances these are the 4 structures, that is the pigments which are present. These are the components present on the thallopoid membrane. These are the very important ones. So these place, what about the photosystem and photosystem 2, that I will discuss later. These components are embedded on the thallopoid membrane, on the chloroplast, and they are facing towards the stroma, and they will be facing so they will be there present on the membranes, only on the membrane layers. Now coming to the photosynthetic pigments. So the photosynthetic pigments are, and the so this one, the photosynthetic pigment. So it is very, very important that that is the structure of the chloroplast, ultrastructure of the chloroplast. So these are the pigments, photosynthetic pigments. Now we will be studying the chloroplast. So in the chloroplast, we will have the different pigments. The one, there will be totally 3 pigments we find. The first one is chlorophyll. The second one is carotenoids. And the third one is phycobilins. So now coming to the major photosynthetic pigment, that is the chlorophyll. Here, especially in case of the chlorophyll, the major that will be, you will have the chlorophyll A and the chlorophyll B is very common in the angiosperm. So the chlorophyll A is the photosynthetic pigment, whereas the chlorophyll B is the accessory pigment. Along with this one, along with these are the pigments present in the chlorophyll. The chlorophyll, the structure of chlorophyll is nothing but is having 4 chiral rings, and a tail. So chiral rings. The chiral rings in the center, you will find the magnesium. So you will find the 4 chiral rings. So you will find the tail. So this is in the center, you will find the magnesium. So this is the structure of the chlorophyll pigment. So the chlorophyll pigment which is present in the plastid, we call them as chloroplast. And also here the chlorophyll B little, that's the one hydroxyl group, that is and these are X-ray pigments. Now coming to the carotenoids. In the carotenoids, there are 2 types are there. The one is carotenes. The other one is xanthophylls. Carotenes and xanthophylls. These are also X-ray pigments. These are all these except chlorophyll A. The chlorophyll A is the chief photosynthetic pigment. In all in the plants, the chlorophyll A plays a very important role for the photosynthesis that we will discuss later in the mechanism. And remaining other are the X-ray pigments. X-ray pigments are nothing but they protect the chlorophyll A molecule. They are accessory. The X-ray means extra pigments are there, and the function of the X-ray pigments is to protect the chlorophyll pigment by solarization, means by photooxidation, means it will get oxidized, not to destroy. The chlorophyll will be, solarization will be due to the heavy sunlight. So because of the maximum solar radiation, the chlorophyll A molecule will be get photooxidized or it may get destroyed. That is, we call them as solarization. So this will be protected by these X-ray pigments, that is chlorophyll B, carotenes and xanthophylls. And especially the phycobilins, they are present in the blue-green algae and also red algae. In the blue-green algae and red algae, these are restricted to the algae, blue-green algae, that is cyanophysian. And these are having, and now coming to one by one, in the carotenes are nothing but they are the unsaturated, and these X-ray pigments, especially these pigments, they are insoluble in water and they are soluble in solvents, that is ether and the acetone. This will the practical you will be studying how the separation of chlorophyll pigments by using acetone. That is, they will be soluble in acetones or there is organic solvent, ether or acetone solvent. And also here the most important one is the carotenes, and the xanthophyll plays a very important role in the angiosperm. Especially the carotenes, usually they will be red or they will be yellow in color. In some cases they will be yellow or they may be brown, and there are different color and orange. So they will have the different coloration due to the different solar spectra. And also there is a very important one is the carotenes. They are the unsaturated, that is they are the small chains of the unsaturated hydrocarbons. Whereas in the xanthophylls you will find many hydroxyl groups. And these carotenes plays a very important role. It protects the chlorophyll molecule from solarization and also from the photooxidation. So if the oxygen bends, the chlorophyll will get oxidized. So it protects the chlorophyll A molecule as compared to the chlorophyll B. Also it protects, and also the other one, the xanthophylls. These xanthophylls are nothing but they are the pigments which are there along with in the chloroplast only, along with the chloroplast. These xanthophylls, they absorb the excess of radiant energy, and then whenever the chlorophyll A requires that energy, they will transfer the energy to the chlorophyll A molecule. So this transfers, we call them as resonance transfer. They will transfer, and these are the important characters of the xanthophylls and the important function of the carotenes as well as the xanthophylls. And moreover, now coming to the red algae, that is the phycobilins are nothing but they are the biliproteins. They are called as biliproteins. And these biliproteins, they are having phycocyanin and phycoerythrin is there. The phycocyanin, which will be bluish in color, phycoerythrin, which is reddish in color, and these phycocyanin and phycoerythrin, they perform the same function as the top in the lower plants, that is in the algae, aquatic plant, lower plants, especially like that is like the carotenes and the xanthophylls. Means they absorb the maximum sunlight, means the excess sunlight, which is not less, which will be absorbed by these phycobilins, and also they store them. Whenever the chlorophyll requires that energy, they transfer them. So these are called as the accessory pigment. So now coming to the photosystem. Immersion. And Lewis. In the year 1943. These are in the last class I have explained you the immersion, who explained about the photosystem. That is about the photosystem, immersions, photosystem. So the photosystem, he was working with the monochromatic light, with a single light with the different wavelengths, that we call them as monochromatic light. A quanta of light which has been exposed to the chlorophyll molecule, quanta means a pocket of energy. So a quanta of energy, he was working with the different wavelengths of the light. And also what he denoted, especially the immersion effect, that is at the particular spectra, the removal of oxygen, that is the bubbling, that is the removal of oxygen that will be taking. So he calculated indirectly. He plotted the graph against the wavelength and rate of photosynthesis. He was calculating indirectly the rate of photosynthesis. He was calculating indirectly in the plants, and he plotted against the graph, that is the wavelength of the light at the monochromatic light at different wavelengths, and also the rate of photosynthesis. So, and as the rate of photosynthesis will be increasing at a particular range, it will be more, and then later it will start decreasing. So yes, the rate of photosynthesis will be increasing in the red region of the spectra, red region. So then immediately at the maximum peak, the rate of photosynthesis will be high. How he calculated that is with the liberation of oxygen. As we are knowing earlier, as we are knowing already, that is during the photosynthesis, the carbon dioxide is reduced and the oxygen will be liberated. And he calculated that is the quanta of light which will be sufficient to liberate a molecule of oxygen will be totally 8 quanta. That is totally 8 quanta. That he calculated. 8 quanta of light is, that is a quanta of light is needed to liberate a molecule of oxygen. Means to synthesize a molecule of hexose sugar, the totally 8 quanta of light is required. That he was there. And especially during the flow, that is when he calculated at a particular place, at a particular region, there is a decline in the rate of photosynthesis. And this he calculated as, that is the red drop effect. Red drop or red drop effect. And also it is called as immersion effect. Immersion effect. So the rate of photosynthesis will go on increasing at a particular region, at a particular in the wavelength, and then immediately it starts declining. This we call them as, at the red region, he called them as red drop effect or red drop effect or immersion effect. And then this was at the 680. It is 680 nm nanometer. The first he observed in the 680, that we call them. And later he calculated with the combined effect, with the different wavelengths, that is the 680 as well as the combined, the short wave and the long wavelengths, he has of them. Then there was the enhancement of the photosynthesis. Then that he called them as immersion enhancement effect. Enhancement effect. So Emerson was the first man to explain about the photosystem. He was the first man to explain about the photosystem. Earlier, people were knowing that, people are calling that is the, in the chloroplast, there is some reaction centers will be there. Those centers are there, are responsible for the photosynthesis in the earlier. And also later, the quanta of light is there. There are some quantazooms are there. Those quantazooms which absorbs the light, and they are responsible for the photosynthesis. But the Emerson was the first man to explain there is the photosystems about the photosystem. And then due to that, there will be not only one photosystem will be there. There will be photosystem. There will be two will be there. Photosystem 1 and photosystem. He explained that when the rate of photosynthesis was enhanced, and he explained that he was able to find that if there are two photosystems in there, the first one is the photosystem 1, and then the other one is photosystem 2. The photosystem 1 that we call them as PS1, and it is also called as PS2. And also it is also called as pigment system 1 and pigment system 2. Both are same. The pigment system 1 is also called as pigment system 1. And pigment system 2. So these are the two, pigment system 1 and pigment system 2. We call them as PS1 and PS2. And the PS1 that we'll discuss one by one, PS1 and PS2. Pigment system, that is very important. Then after completion of that, then we'll study about the mechanism of photosynthesis, means how the photosynthesis takes place in the plants. Now pigment system 1, photosystem 1, pigment system 1. That is composition of photosystems. So this is in the composition of the photosystems. It is a especially it includes totally two photon, that is PS1 and PS2. So these are nothing but it is a mass, it is a group of pigments, different pigments, enzymes, minerals, and all they are present in a system, and in a collectively, that we call them as photosystems. So these photosystems, Yes. Now I will be explaining about the photosystems. What is meant by photosystems? Already that is the composition of photosynthesis. Already the immersion explained, that is immersion enhancement effect. Immersion enhancement effect means there will be earlier there will be only one photosystem, thinking that there is only one system, photosystem. But as there is the after 680 nanometer at the absorption spectra, slowly the rate of photosynthesis will decrease. So then he observed the combined effect of the different wavelengths of the light, and then the photosynthesis will be, the yield will be more. So with that he explained that is there will be two photosystems will be there. Photosystems, the two photosystems PS1 and PS2. The photosystem 1, it will be a spherical ball-like structure which is having 85 nanometer. It is a group of pigments, 85 nanometer in diameter, and the reactions that is it is called as P700. Why? Because the chlorophyll A molecule absorbs at particular nanometer, that is spectra, at 700 nanometer. So for that reason, it has been named as P. About 200 to 400 molecules of chlorophyll A molecules will be there, but chlorophyll B molecules will be less. But along with them, there will be less carotenoids, but more xanthophylls. That is, more carotenoids and less xanthophylls will be there. Along with them, there are other enzymes and also other photosynthetic substances which are present in the photosystem. That is the LHC1, that is light harvesting complex 1, and also FRS, ferredoxin reducing substances. That is also a substance, that is the ferredoxin, which is an iron protein, and also that is plastocyanin. Plastocyanin is a copper compound. That is a copper protein. That is, these are the substances which are present in they are the main substances which are present in the photosystem 1. And in case of the photosystem 2, it is also a group of pigments they are embedded on the chlorophyll molecule, that is on the thalloid membrane. They are embedded on the thalloid membrane. And moreover, these are the antenna pigments, means which absorb the different wavelengths of the light. So that is called as antenna pigments. The antenna pigments are nothing but these are the pigments only. And also here, that is the about 110 nanometer in diameter. And also it is called as P680, means the cloth that is at the nanometer of 680 wavelength, the absorption spectra will get the absorption spectra, that is 680. So it is named as P780, 680. That is called photosystem 680. And along with that, chlorophyll A and chlorophyll will be less in number as compared to the photosystem 1. Along with that, there will be less carotenes and more xanthophylls. So the absorption more xanthophylls. And also there is LHC2 will be there. LHC2 means light harvesting complex. They are the complex of the proteins. They will be there along with the phyophytin. That is the another compound which is called as the phyophytin. Next one is the plastophenylquinine protein, and also then that is the manganese proteins complex, that is called OEC, oxygen evolution complex. So these are all present in the photosystem. That is the photosystem. So it is a model of a photosystem. You can see here, and it may be PS1 or PS2. In this one, we will find these are the structure, the reaction center, that we call them as chlorophyll A, where the reaction center will be there. Different pigments will be there. Different pigments, they are nothing but they are the proteins. These proteins, they will absorb the wavelength, light, the different wavelength of light, and they store them whenever the chlorophyll A is required. So they transfer these energy to that is the successive proteins. That is the one. So this transfer of energy we call them as resonance transfer. Resonance transfer, as I explained you, the transfer that is called as resonance. They transfer to the nearing molecule and finally they reach to the reaction center. That is the chlorophyll A. So these proteins, they protect these are the pigments, they protect the chlorophyll A molecule, which is the chief photosynthetic pigment. In all the algae are different, in all the plants. So that is called as reaction center. So this is about the photosystems. Now, yes, there will be two photosystems, that as I told you, and these are the different pigments which is there. And moreover, and now we'll study the mechanism of chlorophyll, mechanism of photosynthesis. Among them, these complexes, they have that is the iron compounds are there, the complex which will the manganese protein complex, where the Mn and Cl will be there. So this complex protein which will be helpful to liberate the oxygen from the water. And now coming to the mechanism of photosynthesis. Now we will be discussing the mechanism of photosynthesis. Yes. Tomorrow we'll discuss the mechanism of photosynthesis. Today we have discussed about what we have discussed. We have discussed the ultrastructure of chloroplast. The chloroplast is having two membranes, the outer membrane, inner membrane. They both are the double lipid membranes which are impermeable, but they have the carrier proteins inside the membrane, and those carry the substances which are necessary for the photosynthesis. Then you will have inside you will find the matrix, that is the semi-fluid, that is called as that is called the stroma. In the stroma, you will find the coin-like structures or disc-like structures which are placed one above the. They are called as the stroma. Sorry, they are called as the grana. These grana, they are interconnected by a special membrane that is called as grana lamellae or thalloid lamellae. Those are there. And then you will find the DNA of chloroplast DNA, which is circular like prokaryotic. It is having about 1,45,000 base pairs, and it will have the RNA, DNA, RNA, and along with them you will have the ribosomes, and the starch granules will be there, and the stroma will have the soluble enzymes which are responsible for dark reaction of the photosynthesis. That I will explain you what is dark reaction. And also the other enzymes which are responsible for the synthesis of various amino acids as well as the fats, and moreover there will be three spaces will be there. The one space which will be intermembrane space, the second space which will be the stroma phase, the third space will be in the thalloid phase. The thalloid are nothing but they are the sac-like structures which are present in the form of the dishes, petri dish or coins. They placed one above the other. And moreover, and also these ultrastructure chloroplast, and it is having various pigments which are present in them. Those pigments, they are the different complex will be there. That is the photosystem 1, photosystem 2, and also the cytochrome complex, and also ATP synthetase. They are embedded on the thalloid membrane, and they will be facing in the stroma, but they are embedded on the membrane itself. These photos, that what we have discussed, different complex, they are present on the thalloid membrane, only on the membrane, and and these are the important character. And now there will be the immersion effect. That is the immersion what I explained. It is specific. Then he plotted a graph against the wavelength, rate of photosynthesis against the wavelength. At the particular wavelength, the maximum amount of photosynthesis will be taking place. Later it start decreasing. So that is at the 80 nanometer when he was explaining about the monochromatic light of different wavelengths. So and he named that one as photosystem 2 as PS680. So that is the red drop effect. That is the immersion effect. This at the red zone of the spectra, the rate of photosynthesis will be very high, and slowly it declines. Then next, when he observed the combined effect of the rate of photosynthesis, so there will be rate of photosynthesis will be more when it will be having different wavelengths. There is a short wavelengths also that had been added, and then he observed that is the enhancement of the photosynthesis was there. So he called them as immersion enhancement effect, and he was able to explain that there are the two photosystems. They are working in the chloroplast simultaneously during photosynthesis. That is photosystem 1 and photosystem 2. So the photosystems are nothing but they are the required reaction centers with different proteins that just now I have explained. Yes, this finishes in the next class we'll explain about the mechanism of photosynthesis. Yes, thank you. ಬರ್ರಿ ಏನಾಯ್ತು ನೋಡ್ರಿ ಅದು
ನಾನ್ ನೋಡಿ
ಅದು ಮೂರ್
ಇಲ್ಲೇ ಸರ್ ಎಲ್ಲ
ಏನಾದ್ರೆ ಬತ್ತು ಹೆಂಗ್ ಹೆಂಗ್ ಮಾಡಿ ಏನ್ ಮಾಡ್ತೀನಿ ನೀವು